All right, it's 3 minutes past 10, so I think we can get started. Good morning, good afternoon, or good night, wherever you are. Welcome to Speaking of Oats. My name is Pablo Olivera. I'm director of Oat Global. Speaking of Oats, it's a partnership between the Oat newsletter and Oat Global. Charlene White, editor of the Oat newsletter, is with us today, and we'll be handling the question and answers after the seminar. Oat Global is a public-private partnership that aims to coordinate and connect all researchers and oat extension professionals worldwide with each other and with individuals along the entire oat value chain. We are glad you're here today. But before I introduce today's speakers, I want to take a moment to remind everyone that you can use the Q&A widget to ask questions throughout the presentations. Please use the Q&A widget and not the chat function for questions, since this preserves the question for later access. Today's session is being recorded and will be available later via YouTube. Today's speakers are Pekka Lehtinen, research team leader at VTT, and Ulla Holopainen-Mantila, senior scientist at VTT Finland. Today, they will talk about oat as a multifunctional alternative protein source and the Finnish OatHow study, novel quality indicator for food oats. Pekka and Ulla, thank you so much for being here today, and the floor is yours.
Thank you, Pablo. So we agreed that I would start, and then Ulla would continue after my introduction. So my name is Pekka Lehtinen. I work as a research scientist, research team leader at VTT. VTT is a state-owned research center located in Finland, close to Helsinki, and all in all, we are like 2,000 scientists working in VTT, and around the food area, there is something like 80 plus scientists working in the food area, and oats is a kind of a very important topic for VTT and the food research that we are doing, and there is a kind of already like a decade-long history of oat research. So I will today, in my introduction, look at the possibilities of using oat as a multifunctional alternative protein source. And for using oat as a protein ingredient, there is a certain benefits, like there is a very balanced amino acid profile, and the protein has a good digestibility. The flavor of oats is well accepted by the consumer, so majority of the consumers found oats as a well accepted and pleasant flavor. And in addition to protein, there are many other compounds in oats that support well-being, like a dietary fiber, lipids, vitamins, and minerals. And this is now kind of justifying the title of this presentation, the multifunctional. So when targeting oat protein ingredients, it would make sense also to consider the other compounds present in the oats. And major target applications for oat protein ingredients are like dairy alternatives and meat alternatives, and especially in the area of dairy alternatives, we are seeing now quite many oat products. In meat alternatives, there is a quite limited number of oat products using oat as a protein source. And I will have an overview on that on the next slide, which is giving a kind of overview of the global meat alternative markets. And as we know, it's increasing, growing very rapidly. So we are seeing a kind of huge yearly growth rates, over 10%. And there are certain raw materials that are dominating this sector, and soya is by far the most biggest plant source for meat alternatives. Then there are wheat and pea proteins that are kind of, especially pea is kind of increasing, and then fungal or algae proteins, and then other plant proteins. In this picture is this very, very tiny segment, which contains all the other plants than soya, wheat, and pea. And even in that sector, the oats is not the not the kind of very big one. So definitely oat is not the kind of major ingredient in this sector, but there is still a potential for oat to be an important protein source, and there are possibilities for the future growth if there is a possibility to make a good ingredients. And this is the kind of matrix that can be used to evaluate the protein usability or plant protein sources. So there are like factors like protein concentration, then the amino acid balance, allergen risk, commercial stage, whether it's like already the technology is ready for commercialization, or if it's more like in the R&D or startup level. Then there is a flavor sector, functionality, and then of course the production costs, and then also the kind of overall availability of these crop, and this type of matrix has been put up by the now I'm sorry, I don't see the reference in the bottom, but it's the Good Food Institution, I think was the name of that institute. And now we would then look at how oats performs in these matrix. So we can see that the protein concentration, it's not the kind of very big concentration. So there are definitely better sources of protein based on the protein concentration. For example, soya or some legumes definitely contain more protein. Then the amino acid profile oats is quite good. So we can see that there is this relatively high amount of leucine and tryptophan, which are often limiting in cereal or plant sources. There are not major allergen issues related to oats, and in fact, oats is quite often well tolerated also by celiac patients. The commercial stage with oats is not so advanced yet, so we do have a oat protein concentrates on the market, but it's not the kind of a very common product and not available in big quantities. Flavor, on the other hand, it's a well adjusted or well tolerated, so oat flavor is found pleasant by majority of the consumers. Then where oat has a kind of issue is the functionality of the protein. So it's either not very soluble, so it's relatively insoluble, which makes certain applications in dairy segment challenging, and it doesn't form a kind of fibrous structures for meat alternatives easily. So it's not the kind of ideal in that term. Then because of this commercial stage is still kind of early, there's not a good overview of the cost effect. So what is the price of oat protein concentrates or oat protein isolates? And then global crop volume, it's in this evaluation, it has been considered as yellow, but as we know that oats is I think would it be the 6th biggest cereal, so there is a good availability of oats for this purpose if that is kind of converted into the protein ingredient production. So currently the major use is in the feed sector. So then going a bit more into the details of the quality of oat proteins. So as mentioned, there is these essential amino acid leucine and tryptophan, which are present in quite high amounts in oats. So that makes it, compared to the other cereal proteins, it's a good amino acid profile. The digestibility is is quite high, so it's comparable to rice or corn. And as mentioned, oats are often tolerated by celiac disease patients, so there are no major allergenic issues. So then looking at what kind of functionality we would need from oat proteins for different end products or food products. So the foaming property that would be required when replacing egg or other foaming proteins, and an application here could be, for example, cakes or meringue. Then in these plant-based drinks like dairy alternatives or milk substitution, there are two important parameters. It's colloidal stability and solubility. So in this colloidal stability, oat protein is quite good. So we can see that the oat dairy products on the market have quite a good colloidal stability, but the solubility of the protein is not so high, and in fact, a big proportion of the oat protein in these processes is going to this side stream of spent oat grains. Then there is a gelatination performance, so of proteins, and that would be then required in different type of jellies, puddings, or yogurt type products. And then in these meat alternatives, meat substitution, there would be a need for this type of fibrous structure formation. And oat protein as such is not a very good ingredient for this fibrous structure formation, but then the good thing is that we can also affect these functionalities, so we can do treatments to improve the properties of oats. So looking at oat proteins in general, the majority of the protein is not soluble in water. It's these globulins which are soluble in dilute salt solutions, or then with increasing or decreasing pH. Then there is this albumin, which is a water-soluble part that's mostly enzyme, so it can be 10–20% of the protein. And then there are these kind of more difficult to solubilize prolamins or then residue glutenins. Then looking at the performance of oat proteins in different pH. So in this picture we have in red is the solubility of the protein. So we can see that there is a kind of minimum which is around pH 5 to 6. And then if we go to the higher pH, we can increase the solubility, and that can be used in manufacturing of oat protein isolate. So there is often a kind of alkaline extraction step. But also then in the acid pH we can see the increased solubility. So it's a kind of also relevant for some of the dairy alternatives like these fermented or acidified products, or then combination of fruit juice and oats. Then it would be products closer to this pH 4. Then there is also a zeta potential, which is in blue in this figure, and generally this gives an overview of the stability of the particle system in suspension, and typically so that when this zeta potential is close to 0, it's a kind of unstable system. So we can see that also in terms of that this zeta potential, there is a better stability in low pH or then high pH, but this kind of intermediate section around pH 5, pH 6, it's a bit difficult because it's not stable and the solubility is quite low. And this was a kind of evident also on those first generation oat dairy products when they were used in the coffee applications. There was this type of precipitation of instability. So that has been now kind of improved by the use of different additives to change the pH performance. Then looking at usability of oat protein in this dairy segment, it's quite well suited for this application. There is this colloidal stability, and the flavor is well accepted. But the problem, as mentioned already, is that the large proportion of proteins remain in this residue solids in spent oats. But there is a kind of possibility to do the functionalization of these proteins. So we can do the treatments to improve the functionality. And in case of these dairy alternatives, especially improving the solubility, or then if it's a kind of yogurt type product, the gelling performance. And then in the area of meat alternatives, there is also this kind of supporting factors for using oat proteins in meat alternatives, but then there is it's not the kind of perfect in terms of functionality, and it's not very easy to make fibrous structures out of oat proteins. And again, here it's possible then to do this type of a functionalization to improve the protein network and then increase this fibrous structure formation. And this type of functionalization, it can be then done to improve this type of technological quality, like in these examples of dairy and meat alternatives. It can also then affect the sensory quality, so to have a kind of removing unwanted off tastes or kind of textural properties in terms of mouthfeel. Then also the nutritional quality, so there is a possibility to functionalize the protein and other components to improve the nutritional quality. So digestibility, or then, for example, if it's a kind of hybrid products containing both dietary fiber and protein from oats, so it's a possibility to improve the nutritional quality of them both. And this type of functionalization, it can be done in the ingredient manufacturing, or it can be combined with the food manufacturing, for example, in this dairy environment. And the technologies that we can use in this functionalization, it's enzymatic treatment, so we can use cross-linking enzymes to alter the proteins, or then hydrolytic enzymes, or some other more specific enzymes. We can do the fermentation for oat material to improve the technological properties, or then we can do different type of thermomechanical treatments. And this is my final slide, so I would stop here. But there are some ideas on where to focus the development of oat ingredients, and these are also valid, I think, for oat protein. So there is a kind of need to improve still the fractionation technology, so it would be more robust, it would be more kind of easy to adapt the fractionation process, both like a wet or dry fractionation, and also perhaps to include then some functionalization steps. And these functionalizations then, especially looking at the dairy and meat alternatives as a target application. Then, which will be the topic of the Ulla right after my presentation, is the validation of indicators of using oats for food application. So typically we don't have such a good quality parameters for oat products. So it's for example, when compared to the malting barley or baking wheat with oats, we don't have that kind of standard set indicators, quality indicators. And then finally, there is a kind of need then to, if we are producing an oat protein fraction, we should also identify use of other streams like a starch or dietary fiber or lipids, or combine them in kind of clever way. But I will stop here and I let my colleague Ulla Holopainen-Mantila continue with her presentation. So thank you for listening, and I will stay online to ask any questions that you or comments that you may have.
Okay, thank you, Pekka, and thank you for also from my behalf for the opportunity to have this talk on this forum. So my name is Ulla Holopainen-Mantila, and I'm working as a senior scientist at VTT in Pekka's team. And today my topic here is Finnish OatHow study, novel indicators for food oats. And I will first tell you the background of the study and about the structure of this OatHow study we have finished, and then I will give you some result highlights of this study. And I will also have literature listed or the publications of the OatHow study listed in the end, so you will have that as well. But then we deep dive through the OatHow study. So the drivers of the OatHow study, they lie basically in the importance of oats, because it has been use of oats has been increasing, and there's many reasons for that, and big reasons are globally the climate change and also the public health concerns. And because oat, as you know, has many health promoting effects, and in other way, oat could be utilized and contribute to the sustainable food production very well, and has great potential for that. But as the use of oats is increasing, we have been understood that we should understand and have more knowledge about the behavior of different oat varieties and oat crop batches in different kind of food processes, and how the quality affects, how the quality of the raw material affects the end product quality. And this is highly important, for example, if we are thinking of the end product quality, because in industrial manufacture, you are supposed to produce uniform quality very consistently. So one should know the raw material to do that. So the aims of the OatHow study lie on these drivers. So we set as the main aim to develop quality indicators for oats in order to facilitate purchasing of oat batches and to selection of oats for various food processes. And kind of the sub aims we had as we aimed to result in new insights on oats applicability, and also to produce knowledge for breeding of new oat cultivars. And with this kind of plan, we got funding for the OatHow study. And the title, official title of the project was Novel Indicators and Technologies for Oat Quality and Applicability. and as an abbreviation, we used OatHow. So that's the name of the project or study. And this was 2-year project, and it was mainly funded by Business Finland, which is a governmental innovation funding body in Finland. And part of the funding was from the research partners, and to small extent also from the participating industry. And originally this was 2-year project, but due to COVID-19, we had some extension for the project duration, and we finished the project in the end of September 2021. As a research partners, we had VTT as a coordinator, and then we had Natural Resources Institute Finland, University of Helsinki, and University of Turku. And as industry partners, we have 13 Finnish companies that represented oat industry from the breeding and food ingredient production all to the oat-based food manufacture. So we had all the supply chain represented in the consortium. And the budget of the project was 2 million euros. And this is the research setup we had in OatHow project. So we had 4 work packages, which are marked here with the gray boxes. And in first 3 work packages, we went all through from the beginning, from the raw materials, so oat grains, through milling to mill ingredients, and then we did food processing of food models, and ended up also to kind of the oat-based food products. And from every step, we measured different kind of parameters from physical and chemical characteristics of the raw materials and process intermediates and the end products. And we also had parameters from the processes itself. And all this data was then taken to the 4th work package, where we did very comprehensive data analysis of this all data collected from the samples. And we had different sample sets in this project, and here they are presented. So we had one set which had 200 oat samples, and the main set that we were studying was this set of 30 oat samples, and these were all Finnish grown oats. And then we also had 17 international samples, enabling some comparison between the samples. And here in this presentation, I'm focusing on this sample set of 30 oat samples, which we called that we had the processing approach done with. And these samples we acquired 1,000 kilos of each, because they were processed in small industrial scale. And with all these approaches, we aim to result in new technologies and methods for predicting the behavior of oat raw materials in the processes, and to successfully select oats for various processes, and also to enable breeding of high quality oats, and to form quality indicators to facilitate product innovations for export as well. And on this slide, you can see the process scheme we had for the processing of these 30 samples. So here in the upper part, there are the native oat grains, and then there are listed the physical analyses that were carried out with these samples, and then they were first analyzed in laboratory scale. So in smaller scales, they were dehulled as a non-heated oat groats, so there was no heat treatment in this version, and then they were milled as a flour. And here we needed a bit higher amount of sample for the industrial scale. So this was done in 1,000 kilo scale. So we did the drying and dehulling of the grains and the kilning, and this resulted in heated oat groats, and through flaking and milling, we ended up to the heated oat flour. And this heated oat flour was utilized as a raw material for food models. And with these samples, we wanted to evaluate if the measurements that we did from the native oat grains or from the non-heated oat groats, if we could, with these measurements, already predict the properties of this heated oat flour, so kind of the industrially produced oat flour. And here we utilized these physical analyses that are mentioned here, and the chemical composition analysis, they cover analysis of proximate composition, but also very detailed compositional analysis, for example, of avenandromides and fatty acid composition, for example. And we used also kind of a new rapid analysis methods as NIT, near infrared transmission, for this. And we were very happy to see that we had very wide variation in different parameters. And here I have collected some results of the non-heated groats, because we were wanting to have variation in this material, because otherwise it would be very difficult to find interdependencies between the parameters. Largest variation was found in the starch and protein, like it was kind of supposed. Protein content varied between 11 and 20%, and starch content between 47 and 75%. And there was lots of differences in minor compounds, in avenandromides, for example, and also in physical properties. We found differences between the samples. So this was a very good starting point, that we had found 30 samples that were showing good variation in different parameters. Then we observed different impacts of the heat treatment or the industrial milling process on the physicochemical properties of oats. We noticed that after heat treatment, the groats were by their color lighter, more yellow and less red. And also the contents of bioactive compounds were reduced after heat treatment. We noticed that the proportion of damaged starch was increased in heat treatment, and also the pasting properties were changed, and that was shown by the increased viscosity values after heat treatment. And we could conclude, based on this part of the study, that the oat flour properties were possible to be predicted from the native grains. And actually the NIT-based analysis that was quite good in predicting chemical properties of oat flowers. And for example, high hectoliter weight and high 1,000 seed weights, they were related to higher starch and lower protein contents of oat flowers. And we also noticed interdependencies with the color of the native grains and chemical composition of the oat flowers. And these results are already published in this publication. Then I have some research highlights about the food models. So we had food models that represented both or all the kind of products from dry, semi-moist and wet kind of foods. So kind of the moist foods we had was the oat-based milk substitute. And in that we noticed that the composition of oat flour that was used as a raw material was linked to the properties of this oat-based milk substitute. So, for example, if the oat flour with high starch content, that resulted higher mass yield of this milk substitute, and then high ash and protein contents resulted in higher protein content in this milk substitute. And also in these milk substitutes, we noticed lots of variations. So here you can see the least and most stable sample after storage. So there was quite a difference between those. Also in oat baking, we found differences between these 30 samples. You can see the difference in the kind of the best and worst case of the oat breads. So in baking, we used as a model 100% whole grain oat bread, and we noticed that the greatest variation was observed in dough consistencies and bread crumb properties. We also noticed that dough yield optimization was very important quality factor in whole grain oat baking. And there's also a publication about this baking study. Then we did also meat analogs. This was not done with all the 30 samples, but the commercial oat protein concentrate. And this topic was already mentioned by Pekka, as here the enzymatic modification was utilized in order to improve the process behavior of this oat protein concentrate. And here in the images you can see what kind of meat analog was produced with the native oat protein concentrates. So it's looking quite gummy. And here you can see the oat protein concentrate that has been modified with the enzymes. So you can see that after wet extrusion, there's fibrillar structures formed. And we also had dry extrusion food product or breakfast cereal type of product as a food model, and this was interesting food model as we noticed there was differences in the sensory properties in texture and appearance, and also in this dry extrusion food model we were able to actually develop a very new kind of way to simultaneously separate the protein and texturize the protein, and we have done a patent application from this innovation. Then we come to the data analysis. So here For the data analysis, as I mentioned, we took all the data that we had collected from the native grains, from the dehulled groats, from the heat-treated groats, from the flakes, and the flours milled from the flakes, and these model foods. And we had two kind of approaches for this data analysis that was enabled by machine learning, as there was so much data to be analyzed. It would not have been possible with the traditional methods. We had as a first approach association analysis, and then also as a second approach the Gaussian process regression. And these both were aiming to find the interdependencies between the parameters measured and to find if the parameters can be predicted from the earlier process phase. And promisingly, we found that several parameters of oat flour were predictable based on the hyperspectral data of grains with the hulls. So in other words, the hyperspectral data and the hyperspectral data can be measured with a non-destructive way from the grains with the hulls, so that enabled also prediction of the flour that would be produced from the grains. So already that sounded quite promising, but we also noticed that from the flours we could further predict the parameters of the end products like oat-based milk substitute and ripe extrudates. So we were able to identify the target parameters from the various oat processing steps that could be predicted from the measured data measured from the earlier process steps, and we were able to establish a predictive method to provide this methodological basis for prediction of these parameters. As a conclusion of this study, I can say that we covered quite comprehensively the entire oat supply chain. We were able to develop new oat processing technologies, for example, about oat protein functionalization and oat bread making. And all the data that was collected was then taken into the data analysis, and we found that especially that the hyperspectral data of the grains was highly giving good results when predicting the properties of the next process phases. And in overall, we can say that the collected OatHow data it's an essential building block for the future machine learning based indicators for oat quality. And it seems, based on this data, that the hyperspectral imaging would allow prediction of several oat quality parameters. And in the near future, we will be continuing to the development and validation of these preliminary indicators that we have found in OatHow study. So these are now very general and so that they could be utilized in for example, in companies. We would need more data so that they could better serve certain processes. And we have also lots of data produced. So we have, as you did see, we have many publications already published out, but we have still many coming. So we have lots to disseminate regarding the results of this project. And here, as I promised, you can see the list of the publications made in the OatHow. So we had 10 master's thesis works done in this study, and also we have already scientific publications out and many more coming on top of these. And I want to thank you all the project team. It's all the teamwork in such a big project. and I want to thank you for your attention, and me and Pekka, we are waiting for your questions. Thank you.
Great. Thank you so much, Ulla. Also, Pekka, those were wonderful presentations. I can see why you're going to need to use machine learning. Well, you have been using machine learning. That's an awful lot of data, an awful lot of work. And actually, yeah, I'm looking forward to checking out some of those publications. Right now, we currently don't have any questions in the Q&A box. If people on the call would like to ask a question, please type them into the Q&A box. Ah, there we go. There's one from Sophia Marman. Thank you for an interesting seminar. Could you tell a bit more about the genomic approach? What genomic analysis are you performing? Thank you.
Well, actually, we didn't. That was in very preliminary plan to have more thorough genomic analysis in this project, but unfortunately we couldn't proceed with that, and we had with these 200 samples much more lighter analysis with proximate analysis only. So it was kind of the screening of different kind of genomes, to say, in this case.
Okay, well, someone else has asked too, whether you can show the list of publications. One thing, perhaps, would you be willing to send me the list and I can post them in the Oat newsletter, and then everyone can click on the links from that, perhaps after we're done?
Yes.
I suppose you could also show it up here if you like, but yeah, if you can send me the list with the links, then I can post that in the Oat newsletter, oatnews.org for everyone.
Yes.
And Jennifer Mitchell-Fetch says, hi, great and informative presentations. Just wondering if you observed wide differences between the cultivars that were produced internationally, or did their traits and performance fit within the range of the Finnish oat cultivars?
Unfortunately, we, as I mentioned, we had these international samples, and we did some comparison, and they were quite in the range. We didn't notice very big differences, but I think it's fair to say that we didn't have kind of they were kind of how to say, not very representable set of samples, so to say that they were not we just got certain kind of samples, and we were not kind of having very how to say I mean it should have been much more bigger sample set to make fair comparison, and there might be that we do some kind of not good conclusions with comparing these samples, but they were quite in line. We didn't find big differences between the samples.
So are you saying that the reproducibility was low then in those samples?
No, not actually. I'm what I'm trying to find words for is that they were very very kind of samples that we just got from one partner, and we were not able to anyhow say which kind of samples we were wanting to have. So they might be kind of biased somehow. So I think we need to be careful not to say too much, but we can honestly say that there wasn't very big differences.
Okay, right. So they weren't handled the same way as the,
Yes.
yeah. Okay,
Yeah.
great. Thank you.
Are you planning to add more international samples in your study so you can compare with the Finnish ones?
That is a very interesting approach, and if we continue with that, that needs to be very carefully planned, so that there's kind of, for example, certain quality criteria or thinking that it's all food quality barleys or something like that. So the comparison would be fair to say, but as you know, there's so many, for example, in also in Finland, the crop quality changes quite a lot due to growing season, and crop years are very different. So it would need a huge amount of samples to kind of find the variation, even in Finland, and then if we would have more growing sites to be compared. So
when you're growing your oats in Finland, like, I mean, Finland's a very sort of long, narrow country. So how far north, like what's the range in which you can, or which you do grow oats in Finland? Is there a lot of variation in the environments, north to south, or even east to west?
Not of course, there's also from east to west, but from south to north, that's the biggest variation, and oats is not cultivated in the most northern parts of Finland.
Yeah, so I was wondering if that was a factor. One thing too, I mean, over the years, it seems to me that so much of the focus in oats has been, beta-glucan. Everything is beta-glucan, heart healthy, all this kind of stuff. And so, of course, a lot of the breeders have focused on the beta-glucan and all that sort of thing. And so I'm kind of wondering a couple of things. One, should there be more focus now on breeding, well, for protein content as well, maybe even for certain types of proteins, which would have maybe different amino acid components? I don't know. Thinking off the top of my head here. Also, too, do the beta-glucans, does having higher beta-glucan kind of gum up the works? Literally, does that affect how easy it is to extract the proteins and so on?
Do you have, Pekka, idea of this?
Yeah. To my understanding, it's often so that when we are having high beta-glucan oat varieties, that's then meaning that there is a low starch content, and then also the protein content can be high. So often they are kind of going hand in hand, and this happens also during the processing and fractionation. So in terms of processing, the lipids is then another issue that makes it different to process from other cereals, and there's also a big variation in lipid content. So there is a very low lipid and high lipid content and oat varieties, and that when we are having this type of high lipid content, for example, any sieving or air classification will be more difficult or more inefficient than with the materials containing lower lipid content. So it's the kind of combination of all.
Right, exactly.
and still, when oats is kind of main use is still in the feed area, so there is not that much plant breeding activities for example, targeting protein functionality. It's a bit similar that we were discussing in the previous session with the oat cosmetics, that that it's not the kind of big enough driver to target the plant breeding, but I think at the current situation still with the protein functionality.
Mm-hmm. Yeah, and the lipid, that's a good point with the lipids too. I mean, you know, in North America, everybody's breeding for lower oils for, you know, food oats sort of thing. But then you do have some feed oats, and of course the horse trade, they want very high oil in their oats. But I would say, someone correct me if I'm wrong, that most of the oats in North America are actually quite low in lipid content. So yeah, so it might be interesting too. I imagine you're also looking at that sort of thing when you're comparing the international samples to the Finnish ones and so on too. So great. Okay, we have another question here. In the baking portion, you mentioned a small medium particle size worked best. What was the particle size range that you looked at?
I was trying to find it very quickly. I couldn't remember it by heart, but it will be found in the publication more in detail.
Right.
More in detail. Yes.
Okay, that's great. And more on the protein, because yeah, my knowledge of protein chemistry is very, very old and, you know, not current, but also rather limited. So when you're looking at the acid soluble proteins versus the alkali soluble proteins, is there a really big difference in the amino acid composition? Like, does one fraction have a lot more leucine, for example, or tryptophan than the other fraction?
I must say that I don't have that information now available, but the maturity of this easily extractable water soluble protein, it's a kind of enzymes, so it's kind of having the in evolutionary basis, it's like targeting this enzymatic function, whereas the storage protein is then having another target. So probably there is a difference in the amino acid profile, but I'm not able to give there a kind of definite overview. I don't know, Ulla, do you have any information on the different protein classes and amino acid composition?
Not actually.
Okay, that's it. It's curious.
Yeah.
My knowledge is quite limited for a lot of these things. Yeah, so I had another question here somewhere, and I can't find it. Oh, well, that's okay. Does anyone else have a question that they would like to ask here? Oh, yes, there we do. I missed one from Alice Pilgrim. Does Finland produce any naked oat for human consumption? Naked or hullus?
Very little to my knowledge, or do you know, Pekka, in more detail?
Yeah, there is a production, but to my understanding, that's also kind of limited.
Okay. Actually, I see we are actually at the end of our hour. And I do just want to mention, because I did see in the chat that Jay Ohm has mentioned that they are actually still looking for an oat quality scientist at the USDA in Fargo, North Dakota. So if anyone is interested in finding a job there or knows someone who is, he has the link there in the chat. And actually, I can mention that in the newsletter as well. So Jay, actually, if you can send that to me, please. I will put something again in the newsletter. So that's wonderful. Does anyone have anything else? If not, I suppose I should share my slide for the next presentation. And we don't actually yet have too many details for the next presentation. It will be in September. We're taking a bit of a break for the summer, and it's going to be about the Oat newsletter, Oat Global and beyond. So we do want to have kind of a discussion of what people like and dislike and want to see in the newsletter with this, speaking about seminar series, but also in terms of the Oat Global group and so on. And of course, Pablo can speak more to that. Is there anything else you would like to add there, Pablo, for now?
No, I will echo you, and it's next seminar after the summer break or the field break. We would like to hear from the community and see what inputs you have to get our community stronger, and see what do you like to see from us and what we can do together. And to wrap up, also, I would like to thank again Pekka and Ulla for such a great presentation, and all of you for attending the seminar.
And thanks very much, everyone. And we'll see you at some point in September.
That's right. Enjoy the summer.
Or the winter, depending where you are. But yes.
It's true.